In January 2025, scientists finished something that sounds impossible: a fully synthetic eukaryotic genome. A yeast cell with all 16 chromosomes built from designed DNA stayed viable in the lab. That one result quietly changes the baseline. We are no longer asking, can we write a genome? We are asking, can we keep what we wrote stable as life copies itself? Synthetic biology has started to look less like isolated tricks and more like a single, continuous design space. Viruses, chromosomes, whole genomes, even cells now sit on the same spectrum of things you can specify, build, and test. At the small end, the J. Craig Venter Institute demonstrated minimal bacterial genomes like JCVI Syn 3.0 running with 473 genes on 531 kilobases. At the large end, the SC.0 consortium's yeast genome spans about 12 megabases across 16 chromosomes. Different organisms, different complexity, but one shared message. Genome-scale design and debugging is real engineering work now. Yet the counterintuitive lesson is that building the genome is becoming the easier part. The hard part is keeping designed function from drifting away. Evolution does not care about your product requirements. Under replication, selection pressure, and changing environments, cells optimize themselves. They can accumulate compensatory mutations, rearrange DNA, or route around engineered pathways. For leaders used to software updates, this feels familiar, except the system rewrites itself without asking. The SC2.0 yeast genome is especially interesting because it was designed not just to function, but to teach us about stability. It includes LOXY-SIM recombination sites, enabling a system called SC-RAMBLE, which can deliberately reshuffle the genome. That sounds reckless, but it creates a controlled testbed for a crucial question. Which architectures remain stable under perturbation, and which ones collapse into unexpected states? The genome also includes designed elements like tRNA neochromosomes and recoded stop codons. Those details matter because they hint at a future where you can separate essential biology from optional, swappable modules, the way you separate a kernel from applications. A second major driver is cost. DNA synthesis has become routine at gene scale. Commercial providers can synthesize standard constructs for roughly $0.05 to $0.10 per base pair. That pricing turns many experiments from grant-sized to purchase order-sized. Full eukaryotic genomes are still multi-million-dollar efforts, but the trajectory is downward, in a way that echoes sequencing's dramatic cost collapse a couple decades earlier. When the cost of writing DNA falls, the limiting factor shifts to validation. You stop asking, can we afford to build variants? You start asking, can we trust the variants we built over time? That brings us to the biggest operational insight. The bottleneck is moving from construction to qualification. What does qualified even mean for a self-replicating system? For a chemical plant, you qualify a part number. Then you expect it to stay that part number. For an engineered organism, every generation is a manufacturing run, with built-in mutation rates and selection effects. So a serious deployment program needs longitudinal testing. Not just does it work today, but does it retain function after hundreds of generations, under realistic stresses, with measurable degradation rates. This also explains why the most mature deployments remain in controlled contexts. Bioreactors, labs, and tightly managed facilities reduce environmental variation and therefore reduce evolutionary surprise. Field deployment of engineered organisms with heritable synthetic genomes is still limited, largely because containment and stability are not solved problems and regulation remains uncertain. Once organisms can persist and reproduce outside your fence line, governance stops being a paperwork exercise. It becomes an infrastructure-grade risk question. Some of the most consequential work is therefore about making biology simpler, not fancier. A clear design principle has emerged. Simplification improves controllability. Minimal genomes reduce the degrees of freedom that create emergent behavior. But simplification can also increase brittleness. Strip away redundancy, and a chassis that thrives in a perfect lab environment may fail under messy, real-world conditions. So minimal is not the same as robust. Leaders should treat robustness under variable conditions as a first-class specification, not a pleasant side effect. Another frontier is expanding what counts as genetic information. Researchers have demonstrated unnatural base pairs in DNA in vitro, and there have been limited in vivo demonstrations using specialized replication machinery. If expanded genetic codes can be sustained across generations, they offer something strategically profound. A new alphabet can act like a containment layer. If your organism relies on an orthogonal code, it may be less able to exchange functional genes with natural ecosystems, and less able to survive without supplied components. That is bio-containment by design, not just by policy.